Warmly welcome to the session on forest genetic resources for resilient future forests. My name is, as said, Mariana Westergren. I come from the Slovenian Forestry Institute, so just across the border. And I'm really happy that today we have a chance to introduce forest genetic resources and their importance. Because the aim of this session today is actually to raise a little bit your awareness on forest genetic resources. I don't know, but yesterday we had like really nice sessions on one forest and land restoration and on bioeconomy. But actually at the bottom, at the basis of all that are genetic resources, or we can call it also in other words, genetic diversity. And genetic diversity is the basic building block of everything alive. So it's responsible for how fast the trees are growing. It would be responsible for whether a seedling will or will not survive after being planted. It's the basis of resistance of trees to pests and diseases. And I'm really happy that we get a chance to explain a little bit on that. And hopefully at the end of the session, you will all know that, or you will go home and whenever you go back to your work, whatever it is, you will consider or give a thought to forest genetic resources, and you would incorporate them in your everyday work, in your research. And the way how it's going to work today is, first we have a really exciting keynote presentation by Stephen Cavers. Then we will have a panel discussion, a moderated one. I have a few questions prepared for the panelists, but what I would really like is for you to start raising hands when something catches your attention and just ask questions. And the same goes for the online audience. Please write your questions in the chat. And there is a moderator online, Natasha Shibans, my colleague, and she will then read your questions out loud during the panel discussions. And after the panel discussion, we will have 5 lightning talks, so short talks, highlighting some of the diversity of work with forest genetic resources, but also going to different continents. And at the end, all the presenters will have a chance to answer your questions. So now I would like to invite Stephen Cavers from the UK Centre for Ecology and Hydrology to the stage, from Edinburgh, Scotland. And Stephen works with genetic diversity, with gene flow, with adaptation, and how it's organized, and how it arises, and how it affects the functioning on the forests. And his research focuses both on Europe and on Africa. So Stephen, the floor is yours.
So thanks very much for the introduction, Mariana, and thanks to the organizers for inviting me to come and speak to you today. It's great to be back again in person amongst the IUFRO community, and I think it's a unique gathering of people that I think we really need to encourage and do more of, and I'm glad to be back here again. I'm also very happy to see a session on genetic diversity, genetic resources in an all-IUFRO meeting like this. It's really, as Mariana has said, it's something fundamental, and I think we need to keep working to try and get across the message of how important it is that we both protect and make use of genetic diversity in forestry and forest conservation. And so I've titled the talk today, Protecting and Using Forest Genetic Resources for a Changing Environment. And that's really coming from the point of view that we have to protect that genetic diversity in its own right. And that's something that's well recognized now, enshrined in the Convention on Biological Diversity, that we mustn't lose more than 10% of what we have left as genetic diversity. But it's also something that's essential for allowing us to adapt to a changing environment. that we're going to have to make use of that if we're going to be able to sustain the ecosystems that we depend upon for production and for our livelihoods and for survival. So the session today is forest genetic resources for future resilient forests. And resilience is a tricky term. I mean, it has multiple meanings. It depends on who you speak to. The same two different people can see the same system as being resilient or otherwise. But fundamentally, it's likely in most of our understandings of the term resilience that it's going to be delivered by some degree of diversity. And that doesn't mean as much diversity as we can possibly get, but it means more diversity is typically better. And we need to be using that diversity in dynamic systems. And that may be natural systems that turn over themselves, meaning that new genetic variation has an opportunity to enter the population, or it may be in managed systems where we systematically choose and use different types of diversity to get the best results. But fundamentally, we need to be making use of the diversity available to us. And at the species level, that means genetic diversity, the variation within species. And really, the challenge we face is identifying that genetic diversity, figuring out where it is, and finding ways to bring that into use. And so, fundamentally, I guess we still face a bit of not a conflict, but perhaps a division of the ways in how we see genetic variation. That typically, I think we still have a view that's a bit polarized into seeing activity as either conservation at one end or forestry at the other, and not really understanding those as interconnected. And yet, obviously, there's a whole range of different forestry types, forest types and forestry types, all the way from conservation to monoculture forestry. But really, there's a connection between them that's more fundamental than that. And what we really need to try to do, I think, is to understand that as a flow going from one end of the spectrum to the other. That if we can characterize genetic diversity in wild systems, if we can test that, protect it adequately, then we can make use of it in the production systems that we rely on. And that sort of, there is a systematic mechanism for putting that all together and making sure that we get flow from one end to the other. And I just want to illustrate that with a couple of pieces of work from my own group. Firstly, I'll focus on a system that I know very well, which is from the country I come from, which is Scotland. So I'll be looking at Scots pine, but then contrast that a bit with some of the other work that we do in the tropics. So firstly, then looking at this, a case study of Scots pine in the UK. It's one of only 3 native conifers. It's a native species, and it's considered a bit of a national icon in Scotland. It's one of these things that is kind of representative of a sort of a romantic idea of wild Scotland that we like to attach ourselves to. And giving it its due respect, we've spent the last centuries cutting it down and getting rid of as much of it as we possibly can, until we're left now with probably no more than about 10% of what was its original area. And what's left really only survives in a number of very small fragmented populations, some very fragmented. But what those populations represent are naturally selected populations. They've been there. We've as confident as we can be that those populations have been in situ since the species recolonized the UK following the glaciations. And during that time, those populations have had a chance to adapt to the conditions they live in. And we can view those fragmented, dispersed populations as pools of indigenous diversity that we can really be thinking about making use of. And we're approaching that in a systematic way now to try and adequately protect it, expand what's left, and try and characterize and use what we can. So the first stage in that really has been a very systematic effort to identify a series of populations that represent the remaining fragments. So we've identified 21 populations from across the country mapped them, characterized the sort of local conditions that they live in, but have also selected them based on a sort of distribution in environmental space, so that we have as much expectation as we can reasonably have a priori that those populations are likely to be adapted to different conditions. We have worked with the trees standing in those populations and have now genotyped all of them, comparing all of the genotypes from all of the populations. And we can see that there is some population structure. So there is some organization of genetic variation on the ground. And that's likely to reflect small differences in movements of genes through pollen and seed dispersal over time. That means the populations have become gradually slightly different, although as a very highly dispersed species, the differences between populations are typically very small. But then we've taken that the next step. We've sampled from all of the 21 populations, brought seed together, grown them together in common garden experiments, and importantly have looked at those from a whole range of different aspects. So we're not just looking at the kind of productivity traits that might have been typically looked at in a forestry trial in the past, but have tried to extend that more broadly to look at all aspects of I guess, what you would call life history characteristics. So we've looked at growth, of course, but we've looked at reproductive characteristics, timing, phenology of both growth and reproduction. We've looked at disease tolerance traits, we've looked at pest resistance traits, we've looked at the community of organisms that are associated with all of those, with all of the different, I guess, dimensions of the trees, so the leaves, the roots, the bark, and so on, and have related those back to the genetic variation between the trees themselves. Multiple potential traits of value. So there's lots of different ways that we could imagine that we might make use of these different characteristics. So there's lots of value in understanding where the variation is and what we might do with it. And just to give a couple of examples of the sorts of results that produces. So this is a test of Dothystroma tolerance. So Dothystroma needle blight is a common disease of pines and causes needles often to decay and drop. And what we see very clearly is there's a large amount of variation within that population of trees from across Scotland for resistance to the disease. Some showing really quite high levels of tolerance and others really quite susceptible. And that's related to some degree to the population of origin of the trees that we're looking at. So similarly, a more inherent characteristic of the tree, the bud burst timing. We've done a couple of experiments of the timing of bud break in the spring, and we see very clear trends of differences related to the sites of origin, again reflecting the fact that they will be adapted to slightly different environmental conditions in the sites that they grow. And all of this, the very typical patterns, it's the kind of the sort of trends that we're seeing across all of the different characteristics we look at. And all of this reflects the fact that the populations have adapted locally to the conditions they experience. So we've been able to, with that sort of characterization reasonably complete, I think, we've been able to advance now towards actually protecting what stands in situ. And the first step was devising a strategy, which was assembled over the course of the last 3 or 4 years to comprehensively look at the UK's forest genetic resources in a systematic way. That was enthusiastically adopted, thankfully, by the government departments concerned and is now solidly in place. With that, we've been able to advance now to actually getting some populations declared as conservation units. So we've identified sites where the genetic diversity is quite distinctive, where the site conditions are conducive to managing the populations to allow that variation to turn over in a dynamic way. Really, we've been operating within the framework of the U4Gen network, which is a very well developed set of protocols and databases that allow that kind of declaration of populations as conservation units to be made, and then it gives very good guidance on how those can be managed. And in fact, that's been adopted quite widely. So we're quite pleased with the kind of public response to that approach. That there's been a sort of sense of public pride in the fact that we've been able to identify and declare something that's seen as unique variation, and that we've done something about it and we're actively trying to manage it. And I guess the sort of pinnacle of this is that we've actually been able to convince the Scottish government that they should recognise genetic resources as something that needs to be managed as part of an inherent national resource. And they're just in the process of developing a new biodiversity strategy at the moment. And I was very pleased to see that as part of their statement of intent, they recognised the value of having declared the first genetic reserve in the UK. And not only that, but managed to get the language right as well. They actually declared that we're managing it for Scots pine and recognizing that it's distinctive. So that feels like progress, I think. And the final step really is sort of also preparing in parallel a set of ex situ collections. So seed collections are being made from those populations and are being sent to the Millennium Seed Bank at the Royal Botanic Gardens in Kew. And those will be held as a sort of long-term resource protecting what's out there, but also as something that can be made use of for research purposes if necessary. So we've got that system in place. I guess we've got the protection network. We've got it well characterized. We understand a lot about where the genetic diversity is. Can we make use of that? And we've been able to look with tree breeders at what we've got in the tests of genetic variation that we've been undertaking. And just by looking at that straightforward sample from the wild populations, we've been able to show that by selecting for particular sets of families from within that trial, we'd be able to make gains of something like 10% in height growth, 14% in Dothostroma tolerance, which really would be quite valuable gains, and are working now with the tree breeders nationally to feed that variation into the population. And I'm really pleased to say that we've been able to get support now to work on actually directly infusing that variation into the existing breeding program on Scots pine. And I think that change in emphasis, that moving from not just productivity traits, but also trying to bring in other important characteristics in parallel, has really been quite valuable. And it helps to feed back into an understanding of where the value is in that wild genetic diversity. I just also add that in parallel with doing the kind of on the ground work, doing the trait measurements, we've also been developing genomic tools, which we're working towards being able to use to predict trait values. And I think that's really a key thing in kind of accelerating the pace at which we could make use of genetic variation. The ideal will be that we would be able to predict the characteristics of a seedling long before it actually develops to maturity. And that sort of shortening of the breeding gap could really mean a massive acceleration in what we can do in terms of making use of genetic variation. So overall then, I think we've got a fairly good system in place now. We've got a set of conservation populations for Scots pine. They're well declared, they're well recognized, they're protected. We have ex situ collections being made, seed being stored in the seed bank. We've got trials testing the variation that we'll make use of now in feeding into the breeding program. And that ultimately will feed back into material that will be made available for foresters to use in Scotland and the UK as a whole. And really for a country that still has, well, the majority of our forestry, our forest area is still conifer, and the vast majority of that is exotic Sitka spruce. And that's not a resilient state of affairs. That's a very exposed state of affairs. And really, what we're hoping to do with this is to bring in make available material that can be used to diversify the forest estate, give us more species, give us more genetic variation, and hopefully improve the resilience of the forest as a whole in the longer term. So that's where we've got to with Scots pine in Scotland. And to some extent, that's a straightforward situation, I guess. We have a relatively limited number of native tree species to work with. We've actually got a relatively limited forest area where we can characterize that quite easily. But I'd just like to contrast that quickly now with the situation in the tropics. So I work very closely with colleagues in the Kenyan and Tanzanian forestry research institutes with ICRAF and with the Southeast Kenya University. And together we've been looking at a number of different dryland species in East Africa. And really there are a number of other challenges that then come into play. That clearly in the tropics there's a much higher species diversity. So fundamentally there's a whole, a much wider range of things that we need to consider. It's very clear that climate change impacts are much more acute. And actually, there's a higher number, not just of species, but of useful tree species. So there's a sort of a selection question that has to be made. Which ones are we going to work on? And it's a tricky situation because there are very many, there's a long list of species that could be used. There's a long list of species that may not be of so much use, but is, may be of equal importance to biodiversity. But somewhere those choices have to be made. So we've been looking at 4 different species, one species group, a Moringa, Melia volkensi, Acacia senegal, and Osiris lanceolata as species which have value in production. And I just want to focus briefly on what we've been doing with one of those. So the Moringa group is a very valuable set of species. They'll be well known, I'm sure, to many of you in the room. Moringa oleifera is a very widely used species. It has value in its seed, in the pods, in the leaf. All can be used, all are highly nutritious. It can be used for production of oil, which is very highly valued in production of cosmetics and so on. All in all, it has a great deal of interest to people for planting. It's a fast growing species. It can reach over 4 meters in a year. So we have almost instant trees here that really make experimental work quite straightforward, in fact. It's got an interesting characteristic in that there are a number of indigenous species in East Africa, very narrowly distributed, but still used. And then there's a more widely distributed species, Moringa oleifera, which was actually introduced to East Africa from India sometime around the turn of the 20th century. And it's had about 100 years of being grown in situ there to diversify. We've looked at that using molecular markers, so the DNA variation actually shows relatively little diversity, so it was probably introduced from a fairly narrow base, but nevertheless, we've been able to show some differences between different parts of the country, which likely reflect introduction from different sources. Similarly to what we've been doing with the pines, we've established test experiments now using sampling from across the range within Kenya, crossing a number of different environmental zones, and reciprocated those trials in a number of different environmental areas. As with the pines, we've been looking at a whole range of different traits and have been able to identify high amounts of genetic variation in height growth, productivity, pest tolerance, disease tolerance, drought tolerance. And in fact, we'll be extending that next to look at variation in leaf nutrition, leaf nutritional values, or leaf chemistry. But I think what's been very positive and quite interesting is that even from that very narrow genetic base that was introduced to the country, we can find a large amount of genetic variation. So we can consider that as a genetic resource in Kenya's own right. That despite it not being a native species, not having time to, large amounts of time to adapt, clearly over the short period of time that it's been in the country, there's been time for people to select it, it's become diversified, and we see that reflected in the amounts of variation that we find when we bring it together in a common garden. And then the next steps will therefore be to go back to the source populations, identify where the differences are, make sure that those are protected, and then to move on from the initial test trials to develop new and improved varieties that can then be moved out to the population to use for planting. So I will just wrap up then with a few points that really, I think it's clear, I hope, that we can make a link all the way from conservation action to use, and seeing that as a spectrum, as a chain of events really can be valuable. So those genetically diverse populations offer a very high potential for selection. There's lots of variation in it. We see lots of useful traits, and even from situations where there might not have been, there might not actually be very large populations to sample from, there's nevertheless a lot of genetic diversity and lots that could be made use of. And that establishing a more systematic flow going from those wild gene pools to production will not only help us to diversify the production standards that we have, but can also add value to the existence of the wild populations. They will help to secure help to justify the existence of the wild trees. And that hopefully will establish a feedback loop that means there's a clear reason for doing the conservation work. But there's a clear need to test widely to bring these, bring this variation into use. It has to be deployed in lots of different situations. We see very different responses when common gardens are grown in different environments. So understanding how that genetic diversity responds to different growing situations is extremely important. And testing the variation across a whole range of different sites is something that's critically needed. But then finally, I just want to end with a kind of a call, I guess, that there's an urgent need to extend this approach to more species. There's very few situations in which that kind of connection all the way from wild populations through to production is made clearly, I think. And that we need to make the whole process much more dynamic and responsive. We have to be able to, we have to characterize where that variation is, and we have to be able to get out and use it and deploy it in ways that actually mean that we can respond to the changes coming in our environment. And possibly genomic tools are going to be part of that, in being able to predict traits, in being able to predict vulnerability perhaps. But critically, what we have to be doing is testing the variation that we have, finding out what's out there, making use of more traits and bringing that in. It's a tricky problem understanding how to handle areas of high diversity, because clearly, if we're dealing with hundreds of species in an ecosystem, then systematically going through each one, characterizing and testing the diversity that's out there is going to be impossible. So finding ways to use proxies to help us understand where diversity is likely to be is probably part of the solution, but we do need to think about how we handle those areas of high diversity, particularly when it comes to forest restoration, because the common response is just to work with one or a few species just to try and establish a forest. But if we're trying to achieve high levels of diversity, then it's much more difficult. But perhaps that means we've got to balance the effort we make for any one species with its potential use. The whole system of putting in place reserves, test sites, seed supply, and so on might only be appropriate for certain species that have high end use potential. But nevertheless, we may be able to find something in between a sort of, I guess, quick wins that would allow us to use a much wider range of species, much wider range of genetic diversity without the same level of investment. Okay, and I will just finish there. Clearly, this is work that involves a large number of collaborators, both in Scotland and in Tanzania, Kenya, and Uganda. And just obviously wouldn't be able to do any of that without them. There have been a large number of projects that have contributed to this work, and of course, a large number of funders too. So, thank you very much.
Thank you, Stephen. This was really, really nice and very insightful. I hope you feel the same way. Do you? Yeah, did you like it? Yeah, great. So is there any clarifying questions for Stephen? Just clarifying questions. If you want to discuss something more at large and the principles, we will have the panel discussion for that. But just if you need a clarifying question. No, you understood everything. Great. Okay, so Stephen, I will ask you to sit down here and join the panel discussion. And then I would invite Barbara Vincetti from the Alliance of Biodiversity and SIAT, an international organization with headquarters in Rome. Barbara has a background in forest ecology, and she's currently working in sub-Saharan Africa. So with collaborative projects, research in forest genetic resources, how to sustainably use it, how to use them, and how to conserve them. So welcome, Barbara. And then we have two panelists joining us online. So we have Paraskevi Alizotti from the Aristotle. Hi, Evi.
Hi.
Hi. From the Aristotle University of Thessaloniki from Greece. And Evi is an associate professor, works with forest genetics, with tree breeding, with conservation of forest genetic resources, forest reproductive material, and genetic monitoring. And then we have Raquel Diaz Vasquez from Lurizan Forest Research Institute from Spain. Did I pronounce that correctly, Raquel?
Yeah.
Okay, great. And Raquel is the head of the section of research and silvicultural biology and works with genetic improvement programs of forestry species, including breeding for pest resistance. So welcome and thank you for joining us here.
Thank you for the invitation. Yeah, thank you very much.
Okay, so this is the first time I moderate a panel. So maybe it will not go all smoothly. So I apologize for that up front. And I have a few questions prepared for you. And as said before, I would really like this to be an interaction between you and the online audience and of course, our panel. So when you have a question or you want to further elaboration or when somebody has said something, just raise your hand and I will give you a word. And you have seen from Stephen's presentation that when we are talking about forest genetic resources, we are on a continuum gradient from conservation to use and all the all the nuances in between. But my first question to you would go to the breeding aspect, so to the production aspect. Because there is an increasing, we heard that yesterday, there is an increasing demand for forest products, not only for timber, for building industry, for renewable energy, but also for food supply. And, you know, basically the forest genetic resources are the fuel for our bioeconomy sector. So our forest genetic resources, can they provide this increase in demand? With breeding, can we increase that production to what is needed now or not now, but in the future? There will be much more need for timber, for other forest products, for food coming from forests. Can breeding be such a tool that will help us? So I would ask maybe to give the word first to Paraskevi Alizotti.
Okay. Let's keep it short. It's heavy. Okay. So hello everybody. Thanks, Mariana, for the question. And my opinion as a breeder is that breeding can provide these answers and of course the products to forestry. Because up to now, breeding was mainly focused on wood production. But currently, and as the time progresses and the needs for more products becomes more apparent, and also the need for adaptation or plasticity to climatic challenges, I think more parameters are considered when selecting genetic material. So breeding is a very useful strategy and a very useful tool that can incorporate not only production for wood, but production for secondary products like resin, like gums, like fruits production or nuts or whatever, as well as if we incorporate characters that are related to adaptation to stressful environments and climatic challenges. What we need to do in breeding is just to work with selection indexes that incorporate such traits and actually proceed with that kind of material. Up to now, breeding has been focused only on mainly on species that were interesting for wood production. So my view is that there are species that are not studied up now. No breeding efforts have been put on them, but they can be very useful from the point of view of the products that we can get out of them, even from for pharmaceutical purposes and so on. So I think we need to expand our horizons and explore also those genetic resources of other species. And of course, this low breeding strategy is the first one that can be applied in order to pinpoint the genetic material that can be more interesting, and then proceed, if there is an economic basis, to more advanced breeding strategies. I don't know if I can add something more. I don't want to monopolize the discussion.
Please, Evie, add.
Okay. What I wanted to say is that now we have more tools in our hands, because, for instance, we can proceed to methodologies like breeding without breeding, where from natural stands we can just evaluate or estimate genetic parameters and make selections without establishing trials. So there are tools that can be used nowadays in order to make this whole procedure more fast and a little bit more easy. Thank you.
Okay, thank you, Evie. Would any of you want to add something? Because otherwise I have a follow-up question. And that particular would be for you, Stephen, because you work also, and you work also in places, remote places. I mean, we are usually, Evie mentioned that we can have low input breeding. Can this be like something that is feasible in other parts of the world, like outside Europe, outside of more developed world? Would it work?
I think absolutely, yes. Is that working?
Yeah, I think it is working. Can you hear, Stephen?
Yeah.
Okay.
Well, absolutely, yes. I think it can work. I think possibly what we need to avoid is imagining always that the breeding is a complex process that needs a huge amount of investment before it can ever begin. But I think actually you can make a great deal of progress just by going out and selecting good, promising varieties and just getting on with making the choices. It's not something that necessarily has to involve the development of a large-scale breeding program, the development of genomic tools and selection and so on. It can be done in a very low-tech way as well. So I think there's a lot of prospects, and it's just perhaps making the understanding, promoting it more widely that can be done, that could make things move a lot faster in a lot of a much wider palette of species.
So it basically requires somebody to be interested and somebody to be willing to work a little bit with that, and then we can we can get the material, make a seed orchard, and that would work as a low input breeding strategy in other parts of the world? Or is there anything more simpler than that? No, that would be it, right? Okay. And Barbara, knowing that you work a lot with stakeholders and you know, in other parts of the world, not in Europe, I mean, are people interested? Is there like the need in local communities an interest to utilize forest genetic resources in such a way that they would, you know, for increasing food production, for you know, timber, fuel wood? Do you think they would be prepared to do something like that?
Well, I think that when people manage species, they intrinsically also manage their diversity. And there's a lot of, from our working with local communities, there's a lot of understanding of within species variation and properties of individuals and different populations. So there is this consciousness already clearly in the mind of the local communities. And for example, if we look at agroforestry systems in sub-Saharan Africa, we have seen a growing potential in the domestication of wild in species, especially timber and fruit tree species, and a growing interest in farmers in adopting different practices to improve the production and to make use of that diversity that they clearly recognize. So if we look back in the last 30 years of work on indigenous species in Africa, we have seen a first decade from the mid-90s focused on identifying the priority species with most potential, choosing from a very vast pool of diverse potential species. Then we have seen a growing understanding of the characterization of the diversity within species diversity. And in the last decade, we have seen a growing attention to the actual domestication practices that are most successful and interest in upscaling those practices to reach out a larger number of farmers. So I think that there's potential to exploit diversity of indigenous species, which are already recognized as highly value and of high value and already well known by communities for the within species variation.
Okay, that's great. And I assume that the local communities are also aware that they need to protect these resources if they want to use them, or?
Yeah, so I want to link back to the issue of restoring large degraded lands. And I have recently had a lot of experience in working particularly in the Sahel, in Burkina Faso. So we are seeing that a lot of the species with potential for restoration, which are locally appreciated, are rapidly disappearing from the landscape. And even if some of those species are represented within protected areas, we know that if a large portion of the range of these species is found outside protected areas, we have no mechanisms for enforcing any way of safeguarding these resources. So we are discussing with the National Tree Seed Centers, which is already acquiring material from local communities, which are maintaining populations of high interest in different parts of the country. They have already ways to buy seed from the farmers, and they become naturally the guardians of this diversity. So we would like to test the possibility to expand these mechanisms, which is working for just a small range of species for a broader diversity of species. So basically compensating the farmers for protecting these seed sources that are becoming more and more scarce in highly degraded environments under the pressure of deforestation and demographic growth.
Okay, so here clearly we also need a lot of communication, a lot of work on the ground, and also policies, right? Okay, thank you, Barbara. Raquel, and for you, I would like you to, because you are the expert working on breeding for resistance. You know, our forests, it's so much pests and pathogens coming, and the changes in the abiotic stress is only increasing that. I mean, can we find trees that are, I mean, clearly there are some that are more resistant than others. Can we make that, can we utilize that? Can we somehow use that natural resistance to infuse our forestry with that?
Yeah, of course. Well, as you say, there are lots of pests right now, or they can come more pests. So, well, for instance, here in Spain, we have a problem with in Pinus pinaster and radiata with Bursaphalenchus xylophilus, with the nematode, pinewing nematode, and we have with our breeding programs, we have looked for the best trees, the best resistant trees, and we have found some. So right now we are producing the seeds from these resistant trees, and we are trying to establish plantations with this material in the southwest of Galicia, the place where we live, the place where we have the problem already, because it's the nematode is entering from Portugal to Spain in this way. So we are trying to protect the stands with this new material. And of course, I think as we said, I'm sorry, I don't know the name, how to pronounce your name, but she said now the breeding programs, we are starting to have more threats into account, not just food. So we are starting to look for resistance to different diseases and so on, or resistant to climate change. But for instance, I think it's a good thing to do also, that I don't know if we are doing in every place, but I think it's not so much, to establish sentinel plantations in different parts of the world, because these kind of plantations are going to advise us which new pests are going to come into our different places. For instance, if we plant European species in China and Chinese species in Europe, these species are going to be with different potential enemies in these new plantations. So we are going to be able to realize which are these potential quarantine pests or quarantine organisms, and we are going to be able to fight against them before they arrive, to be aware that they exist.
Okay, thank you, Raquel. So that's another really big area for breeding, and where genetics can help to protect our forests. I haven't seen any hands here. Okay.
There are questions from the Zoom, from the online participants. Maybe we can go through them.
Okay, thank you, Natasha. So give me 2 questions from the online audience, if there are 2, and then I will take the questions from the room.
Okay, so the first question that I see here is from Faye, asking if someone can give the opinion on using cloned or asexually propagated seedling material for reforestation purposes. And the second question here is from Christophe, asking, you talked about diversify species. What is the acceptance to exotic species in UK? What do you think about the statement saying we do not make the most of the interest-specific diversity to address the adaptation to climate change?
Okay, so Stephen, I guess the second question is for you, and the first one I will let a volunteer answer. Okay.
I volunteer for that.
Okay, thank you, Evie. So Stephen, go ahead.
Okay, I'll answer the second part then. I think there's, well, it's a very, I would say that the general opinion in the UK is very much in favor of making use of exotic species. I don't place myself in that community. I think I would like to see much more emphasis on making use of what we have as native species. But we've had a long history of making use of exotic species. And in fact, most of our forestry is exotic. I guess that culture means that we're very open to the idea of planting new things. And already we're seeing For example, efforts to try and combat things like ash dieback, which is affecting ash species across Europe. That one of the solutions being widely proposed in the UK is the introduction of exotic species and crossbreeding with our native ash to try and produce something that might resist the disease. But on the other hand, we are already seeing in affected ash populations that there are some very resistant individuals, and it seems that, to me, it seems obvious that that's variation that could be made use of to restore native ash without having to introduce something exotic. So it's a very polarized debate, I think. There's a lot of discussion backwards and forwards, but yeah, I would say the consensus is probably in favor.
Okay, thank you. I think personally I would take your side, but yeah, I also like the native species. So I think we have a lot of variation that we can still use, and especially with the ash dieback, there are trees that are resistant or partially resistant, and we can look into that. Please, Barbara, you want to add something?
I want to follow up to what Stephen was saying, because this is also an issue that is coming up a lot in my job in sub-Saharan Africa, trying to push for having native species more represented in restoration efforts. And we are trying to propose portfolios of species, fully characterizing together with local communities what are useful species and for what they are useful, not just picking from the literature, but also understanding how farmers see the potential of these species and developing scoring system to be able to choose based on restoration objectives and also the stress factors that operate on the restoration side. So with this scoring system, you are already able to come up with a portfolio of options that are reaching multiple objectives, because another challenge is to be able to combine restoration, conservation with use, and you want to achieve the most on the same land. So we are trying to develop these catalogs of species, and we are also trying to promote native species, but there's a major challenge. The tree-seed system behind the nurseries are not able to cope with the potential demand that is expanding in the direction of native species. So we are also trying to support the governments in identifying the major gaps in the tree-seed supply system and boost research to be able to cope with the potential increase of interest around native species.
Could I make one, well, maybe a couple of other quick points? It's just that the list of exotic species that's been tried in the UK, the list of species that have been tried and failed is much, much longer than the list of species that have successfully been deployed. So that's one instance. There's a second issue in that we've got clear cases now documented where an introduced species was introduced with a pathogen strain that then spread to native species. So, there's risks on that side as well. And I think on the other side, that a lot of what we're looking for is added value from the forest restoration efforts or tree planting efforts that are being made. And if we can make use of native species in those efforts, then we're getting a biodiversity return as well as the carbon sequestration potential, the production potential, and it is trying to achieve that balance, I think, between the different objectives that really means, to me, weighs in favor of the use of natives.
Okay, yeah, that's a good message, I think. Thank you, both of you. Evie, please, the first question.
Okay, first a little bit, I would like to go back to the question on whether breeding can do things about resistance to pathogens. And I would like to remind everybody, and of course, I'm sure Raquel knows about that, about the American chestnut and the problem with the cancer. So hybridization among species and breeding efforts towards this direction make possible the incorporation of resistance genes from the Chinese and the Japanese chestnut species. And now the American chestnut is returning back to the American ecosystems, which was a majestic tree. And actually with the bud roses, they are bringing the same phenotype and the same quality of wood and nuts. So breeding has a lot of potential and has a lot of solutions. So there is a lot to do in this part, even in the resistance part for pathogens or whatever. So let's go now to the question that I'm going to address is may I know your opinion on using cloned or asexually propagated seedling materials for reforestation purposes? So my view on this topic is that we should follow a different approach if we're discussing about reforestation and keeping the genetic variation of the forest as big as possible. And there is another approach that we should use if we're discussing about production plantations. So if we're talking about production plantations, then yes, we can use cloned or sexually propagated material, but still not one genotype or two genotypes. So we need to use, even in this case, a mixture of genotypes in order to reduce the risk of a failure. So if we're discussing about reforestation, we prefer to have as much variation as possible to the forests we create artificially. So in this case, we don't need to have cloned or sexually propagated seedling material, as it's going to be limited, the number, and actually the variation is going to be limited regarding to what we will have if we have a representative sample of the genetic variation we need. Let's say from how many seed stands we're selecting, from how many trees within a stand. So we need to have a representative sample of mother trees, collect from them, raise seedlings, and bring back all the genetic variation back to the forest we're establishing. So it's two different things, and I would like to give one example, for instance, for the plantation, for the production plantations and the forestry that really takes place in the eastern part of the United States, where actually the plantations are including only 10 full seed families that are asexually reproduced. But those plantations are for production purposes only. So they need specific material, specific wood with specific properties, with specific resistances, and so on. If we talk about the natural forests of the eastern coast of the United States, then this does not apply. Thank you.
Thank you, David. Yes, please, Raquel.
Sorry, thank you very much. Yeah, I agree with you, and I was thinking that for resistance in chestnut, for resistance to these problems, we have a program here in Lovitan, and my colleagues are working, searching for hybrids between our species and the Asian species to search for rootstocks which are resistant to this disease, and then we can graft the chestnut which is going to produce nuts. Or yeah, so it's I completely agree with you that it's really important, and it's the only way sometimes to fight against these diseases and to have the production we need to have for, in this case, for fruit. In relation with the clones, I also agree with you because it's clear that if we are talking about the main commercial species, the industries and the sector, the forest sector, the owners or silvicultures are going to want these clones or this selected material, which is going to grow better, to have more wood or more any other product. So they are going to want this material, and it's normal. We have to understand that. But of course, it's going to be a problem. So I always would prefer to use maybe parents of families, for instance, than clones, because it's going to have more variability. But at the same time, if there are any clone that is going to have a very much genetic gain than the rest, they're going to want to plant that material. So maybe another thing that we can do is to use small plots, each one with one clone, but a mixture of plots. So maybe silviculture treatments can be used in order to increase a little bit the diversity of where we put all these plants to try to fight against this situation or this monoclonal thing, no? But of course, if they are not for commercial issues, we should use diversity as much as possible. Sorry, as possible.
True.
Yeah, thank you, Raquel, and I'm really happy that you brought up the link between the genetics and silviculture, because silviculture
they must be together.
Yeah, they must be together. Yeah, no, you are really right about that, and I hope that there will be more links in the future. So I had 3 hands from the audience now: the gentleman in brown, then it was Santiago, and then Dave. And please, if it's a question for a specific panelist, just point.
Yeah, thank you very much. I'm a silviculturist, and I have 2 questions. First question to Mr. Covers. You have shown very nicely how the phenotypic traits vary between the different populations in Scotland. Did you find the genes which are responsible for that traits? And the second question is to the panel. There are many tree species, for example, Pterocarpus, a rosewood species in West Africa, and also many tree species in mangrove forest ecosystem. They will be very difficult to regenerate, even if you have a successful seeds, because they need specific growing condition, specific animals, specific microclimate, and so on. And what is the role? Because we are losing those species, some of them are under threat of extinction. So what would be the role of plant breeding and genetics in for such species, which needs a unique condition to grow and to success in the whole life cycle? Thank you.
Okay, so Stephen, would you want to elaborate on answer the first one?
So yeah. I guess the easy answer is no. So we have a lot of genomic information now, and we've clearly got the trait variation, but the more complicated answer is that most of these traits are underpinned by a very large number of genes, possibly all genes operating to some extent, and it's very small differences between them that make the trait differences that we see. So at this stage, it hasn't been possible to clearly identify a set of genes that are involved in explaining trait variation. And typically, I guess, the approach now is to try and use as much of the genome as possible every time in trying to explain a phenotypic variation. And to some extent, that's what's allowing us some success in being able to predict traits. But being able to pin it down to a few genes that we might be able to sort of transfer to individuals is just, I think, is probably not going to be the case for these sort of species.
And the second question, you will really like our first flash talk, because it's about the mangroves with Miss Analyn Pasagdan. But if anybody wants to elaborate a little bit on this kind of difficult conditions, if breeding can help, or it's again the same question that appeared before. Other parts of the world, not so much resources, difficult conditions. The biology of the species maybe is too difficult. I don't know. Anybody feel safe in answering this question? I don't know the answer myself, so.
Mariana, could you please repeat the question a little bit?
It was about the breeding of, can breeding be a solution for the species that grow in, they are very low in density or live in very difficult conditions, like the mangrove, where the biology of the species may prevent such breeding and things like that. So Barbara has.
Well, I just wanted to react to remind a few aspects of the context in which I'm, for example, operating. So the technical solutions that we identify are minor compared to other factors that are affecting restoration or bringing back species in the landscape, which are of a nature and of a caliber that is totally out of scale with the attempts to really maintain a few species that are under threat. And for example, in the Sahel, we have major pressures which are linked to security, desertification, and we don't have, and restoration is conducted successfully if the land tenure regimes is clear, land tenure is secured. In our case, sometimes in West Africa, tree tenure is disconnected from land tenure. So we have a massive pressures on the system, and for some species which are maybe of high biological value but are less maybe appreciated by farmers or less useful in a farming system, maybe we have to be happy with maintaining some relic populations in protected areas or identifying hotspots of diversity where we can put some forms of protections, maybe informal, finding new arrangements of conserving outside protected areas. I think this is the way. There's so many other species maybe that are also fulfilling livelihood objectives that are maybe having priority. So realistically, maybe we will have to do with what is left and try to safeguard what is left.
Okay, thank you. Then we have roughly 5 more minutes, so I hope we manage 2 more questions. Please, Santiago.
Thank you. I just wanted to make a first question. Well, it's not so first question, but like just to be optimistic. So like I see very clear the role or low input breeding and tree selection to extend work from just a handful of species that we are used to work in Europe or North America to the huge diversity you find in tropical regions. But do you see also a role for genomics?
Please, Stephen.
I think in some cases, I think that's and I think increasingly as the technology becomes cheaper and more accessible, that it will probably be more and more widely used. I think probably at the moment, the investment required to develop genomic tools for every new species that we want to improve probably puts it beyond, it's certainly not the answer to rapidly accelerating improvement in a wide range of species. I think we could probably make faster progress just by going out and making some, low-tech improvement, and we'd get further faster if we did it that way. But I think in the future, by all means, when we can have a genome for $10, then we're going to be we'll be able to move quickly.
Yeah, that's heaven for our geneticists. Huh? Okay, please, Dave.
I'm Dave Chakraborty from the Austrian Research Center for Forest. Stephen, I really liked that the Scottish government's intent, it was mentioned about the genetic diversity. So I'm curious to know about what is the overall picture in your respective countries, including Scotland and everywhere, about the trade and utilization of the forest reproductive material, because this is one of the biggest challenges, because we forget about the non-native tree species, but we are not allowed to relax or adapt our guidelines or rules about our fixed seed zones. We cannot bring seed zones which are, we know that this is an adapted seed zone, but it occurs cross-border, but I'm not allowed to bring that. So I would like to know your opinion of all the panelists on this.
It's a really interesting question. And do you mean in terms of international trade? Yeah, cross borders. Well, speaking as someone from a country who's just managed to erect a whole new series of boundaries around itself, we may find that that's certainly, in our case, it may be that it's easier to bring new materials in, although we're also looking at, there's a lot of concern around plant health, what's transported along with reproductive materials. But I think maybe the other aspect is to ask, where the need is. That, for example, the bit of the Scots pine range that we're dealing with in Scotland is a tiny part of a huge, a transcontinental distribution. But actually a lot of movements from the continental distribution into the UK have not been particularly successful, just because the environmental conditions that the two different types have grown in is just too great for it to succeed. So, to some extent, that's where a lot of our emphasis has gone into looking at what variation there is in the indigenous population, because if we can make use of that, then the need for transboundary movements of forest reproductive materials is much less. And possibly that's a useful takeaway message, that actually there probably is variation there to work with locally if you can. But certainly, yeah, there are a lot of issues in terms of moving across borders.
Thank you, Stephen. And that brings us more or less to the end of our panel discussion, unless if one of the panelists wants 30 seconds to add something to this last question. I'm pretty sure some have opinion on that, no? Okay. Then I would like to thank you both, the online panelists. Please stay tuned for the end.
Of course.
Yes, and thank you, Stephen, and thank you, Barbara, for sitting here. It's probably not too comfortable, right? Okay, but just if you can go off the stage, and I will now ask one by the other. After the other, the flash talk presenters, you will have 4 minutes for your presentation, and we will reserve the questions for the last 5 minutes of the session. So please, Analyn Pasagdan, I hope this is correct, your name, yeah? From the Philippines. She will be talking on the inventory and assessment of superior mother trees of mangrove species.
Okay, good morning, everyone, and good morning to our participants online. So I am Analyn Pasagdan, and I'm from Philippines, and I'm going to present our study on the inventory and assessment of superior mother trees of mangrove species in Pagbilao Mangrove Experimental Forest in Quezon, Philippines. Okay, so why did we do it? Philippines is one of the 17 mega diverse countries in the world. We are also a biodiversity hotspot. And out of the 80 mangrove species in the world, half of it can be found in the Philippines. We have 47 mangrove species. But these mangrove forests in the Philippines are really declining at an alarming rate. So from 90s, we have 500 hectares. But now in the 2000, we have around roughly 170 hectares. So almost 70% of the mangroves in the Philippines have been really destroyed. So why? Because of this, there is a need to reforest the denuded mangrove area to sustain its productivity and its genetic resources. Why genetic resources? Because out of those 47 species found in the Philippines, 2 of these species are already threatened species based on the IUCN status. And also one of the species is considered as endangered species. And because of this, we need a high quality planting materials to repopulate the mangrove denuded areas with this kind of species. And with our gentleman here from India, right? Yeah. I agree with him, with his questions, that it's really hard to have a high quality planting materials for mangrove species. Why? Because mangrove is a unique species. It has unique characteristics. It has a zonation, and it is really hard to have quality planting materials. And this, for our study, we focus on, okay, so, sorry. Okay. So for our study, we focus on the assessment of superior mother trees. So this is the Pagbilao Mangrove Experimental Forest in the Philippines. You can find it, you can found it in the southern part of the Luzon area in the Philippines. This is a protected area with a total of 145 hectares. So in this 145 hectares mangrove forest, we have 29 true mangrove species and 19 associate species. So what we did was we conduct a survey. So each mangrove stand was assessed using the selection criteria developed by our office. So this selection criteria is a scoring system. So if any one of you is interested on the selection criteria, you can approach me later. So those mangrove stands were assessed by different parameters. So it includes the tree health, the age of stands, and many more. So those identified superior mother trees are geotagged and labeled for our monitoring purposes. Okay, so as a result, during our assessment in the area, we established a total of 115 individual plus trees or mother trees. So you can see in the screen, we have a total of 24 species of mangroves, so comprising of 13 families. And each species of mangroves, we have 5 individual plus trees, except for the number 17, which is the Soneratia obata. This is one of the nearly threatened species. We only have 2 individual plus trees of this one. So a total of 115 individual plus trees. So in the screen, you can see those pictures of our identified superior mother trees. Sorry. Okay, so again, after identifying those individual plus trees, we geotag them. So in this map, you can see those little points, color-coded points based on the species. So that is a total of 115, sorry. So the established IPTs now serve as sources of planting materials in our area and also germplasm of those 24 mangrove species. So this is the mother trees where we collect seeds of mangroves, propagules, and then we conduct some sort of treatment on the seeds that we collected. And we use these seeds in our production of planting stacks. So every quarter, we have a target of 3,000 planting materials of mangrove. So yearly, we have around 12,000. So those planting materials were then raised in our nursery. We have a nursery in our area that can accommodate to 15,000 of planting stacks. So those planting stacks, when they are already matured enough or grown enough, we have local partners in the community, or we have different stakeholders that request for this kind of planting materials for their activity for rehabilitation of mangrove and also for enrichment planting. So next, to ensure that these mother trees are really still quality, we conduct monthly monitoring and assessment of these mangrove species. Now, the Pagbilao Mangrove Experimental Forest that does not just serve as the source of planting materials, but also serve as a gene pool of mangrove resources in the Philippines. Thank you.
Next speaker is Santiago Gonzalez-Martinez, coming from France, and I will not repeat the title again.
Thank you, Mariana. Good morning, everybody. I'm going to give some highlights about the work we are doing using genomics to try to improve predictions of population performance in natural populations. And this is mostly the work of a PhD student called Juliette Achambo. So if you are interested in getting deeper in these studies, you can always go with her PhD that is available fully online. Okay, I had to get a little bit closer. Sorry. So as I said, like I'm going to talk about the use of genomics to improve predictions of population performance of forestry in natural populations. And this is the work of a PhD student called Juliet. And her work is available online. So if you are interested, you can get deeper by reading her PhD. So this is the general context. This is here in Europe. But there are similar programs in other regions of the world. So we are expecting to do a massive effort in reforestation in the next decade. So, and the question is not, this is very good, of course, and the question is where to plant all these trees, but also what materials, which species and which provenances we will use. And for that, we believe that including genomic data into this decision making could be useful. And in particular, when we are talking about predictive modeling. So, and there is an advantages of using genomics, like with respect to using only climate or phenotypic data. One is that it helps us to understand better the drivers of the process of genetic adaptation. It helps to integrate this very huge interspecific variability that we have within a species that is very often neglected. It helps also to do better predictions when we had to extrapolate outside of the range of these provenances that have been tested in common gardens. And finally, it can accelerate the process of decision making. So this slide is only to show our case study. So we are using maritime pine, that is a very common species in some Western Europe, for which we have many common gardens and some genomic resources, like a model species to learn to how to do this, so we can apply it more widely in other species. And I will show today just a single result of this work. Like here you can see how by using genomic data, we were able to establish some correlations between genetics and environment that allow us to do predictions of what would be the regions, the populations, or the distribution of these species that may be maladapted or have some kind of risks to not be adapted to the future climate in the regions where they are. And thus it has been a combination of genomic and environmental data. So it gives us, it's not perfect, it has several limitations, but gives us an overview of which regions they could be more in danger in the future, and also give us an insights about how, which populations could be useful to be planted in these regions without going to other species, just harnessing the variation we have within the species, but in different regions of the distribution. And thus, we believe it will be useful for the future. And we are very willing to work more with people in silviculture and in reforestation programs to help doing these choices that we know that are hard. And with this work will be continue in a new project that will be starting very soon. And with this, I would like just to finish my presentation and give you all thanks for attending this talk. Thank you so much.
Thank you, Santiago. That was really nice. And you can see that genetics can help or is at the basis of the decisions in restoration. It should be at the base of the decisions in silviculture and many other fields. So with that, I would like to invite our third speaker, Ronan Cashell from Ireland, and he will be taking us deeper into using genomic for improved growth in Sitka spruce.
Morning, afternoon. Good afternoon, everyone. I'll be talking very much through the production side of using genetic resources, though with a mind towards conservation. Sitka spruce is central to forestry in Ireland, for better or worse. It is 51% of our forest inventory, and our forest inventory is 11% of the land cover. We have been breeding Sitka spruce in one form or another since the 1980s. That breeding itself a follow on from my Eufro provenance trials in the 70s. And through that time, we've managed to overall assessments of the breeding population, one for a very juvenile height, another one for a slightly less juvenile width. And the project kind of, those were not synchronous. They happened from between the 1980s to 2007. The project entered a little lull after that, but with cheaper and more readily available sequencing strategies, there's been a kind of revitalization in the interest of breeding, with an idea to do it from the perspective of being genomically led, being guided by genetics. And to do genomic selection, we need two things. We need phenotypic data that relates to our traits of interest, our improved growth, and we need relevant genomic data that can be linked to these phenotypes. Our phenotypic data, we draw from those two earlier assessments. What we had was phenotypic measurements of half-sibling progeny derived from their maternal genotypes, which act as seed orchards. We measured, I think, we have in the range of, 10,000 measurements, each of them relating to one of 215 seed orchards. We got those measurements, we modeled them to create a predictive value that relates to each of those 215, to create a breeding value for both height and width relating to those. And for our genotypic values, we use genotyping by sequencing, which is a very non-specific strategy. You go in, you sequence, you come out with genomic data. It's useful for species that are less characterized maybe than some core species, but combining all that data, you don't necessarily get the same data across all the species. You can have high amounts of missing data. So combining those two data sets, we have our breeding values and we have a genomic data set of roughly 78,000 biallelic markers, with a threshold of 15%. I can go through that choice more later if anyone's interested. And we endeavored to look for the best way to predict for breeding for our traits of interest within our set. We looked at 6 models. The first two there, GBLUP and RKHS, look at the genomic relationship matrix. We can just think of essentially a measure of how genomically similar one individual is to every other individual in the data set, their relatedness. It follows on from pedigree-based breeding. And then the other 4 look at each of the individual 78,000 markers we have and assign a value to each of those that it may additively help or hinder that trait of interest. Our RBLUP is through likelihood, so its answer is going to be the same every time you run the model with the same data. The other 3, the Bayesian, aren't. They use Monte Carlo Markov chain. I won't get into it, but you can think of it like pulling cards from a deck sampling, which is where the Monte Carlo bit actually comes from. And to quickly talk to our results, we have roughly maybe around a mean of 27% accuracy for our juvenile height, less so 1920 for our width. These are somewhat smaller than we'd expect from other similar studies in the likes of white spruce and Norway spruce. It could be our unconventional phenotypic data. It could be gaps in our genotypic data. But we have evaluated that we can successfully breed for improvement. But what's more, we can kind of take this forward. We can breed for other traits of interest. We're currently investigating whether there is the potential for resistance to 2 pests that are prevalent in Ireland, as well as drought within the population we have. We can also hopefully, as we move towards talks of greater afforestation, we could sustainably intensify within the forest region we have. We could move afforestation then to cover our more native species, or just a more biodiverse spread of species. And I'd like to thank you all for listening and all my project partners with helping generate the data under the Genesis project, which this research was done under. Thank you.
Thank you, Ronan. That was really, really interesting. I mean, at least for a geneticist. So now we have a talk by Manisha Thapliyal, and it will be a recorded presentation. So Manisha is with us, and she will be hopefully able to answer questions if there will be any later.
Good morning, good afternoon, and good evening to all the delegates who are attending this conference in the offline and online mode from different parts of the world. I will present about the program on conservation of forest genetic resources of Himalayan state Uttarakhand nestled in the Western Himalayas and the program's linkage to species restoration. As we know that the forest genetic resources mostly comprise of trees, shrubs, and woody lianas, and more than 18,000 higher plant species have been documented from India, and 80% of which is higher plant diversity contained in the forest habitats, and more than 7,000 of these constitute the FGRs. For the program, the species were prioritized and selected on certain criteria like their ecological, socio-economic importance, threat perceptions on the population of the species, the lesser known status, and endemic species to the region, some of them like Pictosporum eriocarpum, Butia pellita, which have highly restricted distribution and only few individuals remain in nature. The program on conservation of FGRs encompasses preparing a comprehensive inventory with population and threat status of the FGRs, preparing their eco-distribution maps, long-term storage of the processed seeds of 100 species in National Gene Bank, studying and evaluating the genetic diversity and population structure, and establishment of field gene banks of important forest genetic resources near the forest areas. The program on conservation of FGRs encompasses preparing a comprehensive inventory with population and threat status of the FGRs, preparing their eco-distribution maps, long-term storage of the processed seeds of 100 species in National Gene Bank, studying and evaluating the genetic diversity and population structure, and establishment of field gene banks .
Okay, thank you, Manisha. I hope you can hear. I hope you can hear the. Oh, it's not finished yet.
Yeah, I'm here. For the long-term conservation of the processed seeds, the seeds of species were collected at optimal maturity from the identified seed sources. The seeds were cleaned, processed. Their quality was, that is the initial viability, moisture content was determined, and then they were desiccated gradually to the safe moisture levels. And then they were packaged as per the standards of conservation of the samples in the gene banks with all the passport data, and then they were deposited in the seed bank of National Bureau of Plant Genetic Resources. Field germplasm banks of several prioritized species like Taxus baccata, Cinnamon tamala, Diplochnema butyracea, Rhododendron arboreum, Myrica esculenta, practices species, and so on have been established under the program. The material that is of these species is also being multiplied further for producing the planting stock of the species. These plants will be planted back in the forest areas, that is in the native range of the species, in close collaboration with the forest department. Due to the restriction of time, these were the important highlights of the program that I could present. We acknowledge the funding support for this study from Ministry of Environment, Forest and Climate Change, Government of India, and the UFRO and organizers for providing us this opportunity to present our research work before the forestry science community. Thank you very much.
Thank you, Manisha. I hope you can hear the thanks in Zoom. And now we have our last lightning talk speaker, Christophe Orazio from France. Christophe, are you there?
Yes, I am. I don't know if you want me to present live or if you want to use the recorded presentation.
No, please, you use your slides.
Okay.
Okay, great. Go ahead, Christophe.
So, do you see my screen?
I think it's coming, not here yet. Okay, it's here. Great.
Okay, so I am Christophe Orazio, the head of European Institute for Planted Forest. I will introduce you a project we have to set up a plantedforest.org website, a resource center dedicated to planted forest. Many of you could notice that planted forests are in front of the scene now for many reasons. The best reason is well known by foresters, is that planted forests contribute highly to the bioeconomy. With less than 7% of the total forest area worldwide, we produce about 40% of the world timber. These are data from 2012. Historically, planted forests were widely used to stabilize soils, especially on slopes. So here you have pictures from 1930 in French Alps. Planted forests can also be used to ameliorate rain runoff, because when you stabilize slopes, you also avoid big floodings. Planted forests can be used also to restore fuel resources in Africa, and not only fuel, as we have seen this morning, we will also need timber for construction. Planted forests can be used to restore devastated forest area after storm, fires, drought. The main issue at the moment in Europe is bark beetle attacks, as you can see here in Germany. Planted forests can also help to quickly restore biodiversity following intensive farming or deforestation. We have many examples in American countries. Planted forests can be used for carbon sequestration. I don't need to develop this. This is well known with the LULUCF process, where we count the total amount of afforestation scheme in each country. But planted forests are unique because they rely on a planter decision. The first decision is to decide what to plant. And this is why it is important to know well the genetics and to have a good genetic resources management, to have something that is suitable for the site. But there is new challenges. One of the new challenges is climate. In the past, when we had tried the appropriate resource genetic resources, we knew what will grow well, but now we have to consider what will happen in the next decade. There is also market uncertainties, energy crisis, that makes the decision of the planter quite tricky, not knowing well if he has to go in a short-term planning or long-term planning. Social acceptance also is making the decision of the the choice of what to plant more tricky, especially as there is some issues with exotic species or the landscape generated by plantations. Emerging pests and diseases are also an issue. We have talked a lot about them this morning. There is more and more breeding program considering the pests and diseases, and so this can influence the choice of the species and the plantation material. For example, who really want to plant ash at the moment in Europe is so uncertain. How to plant? Again, there is many questions here that have to be addressed. You want to plant pure mixed plantation? How do we do in harsh conditions? Oops, sorry about that. How do we maintain and make sure that we will have the reproductive material we need if we initiate big plantation programs? What are the choices that have to be done for breeding programs? How to select the best materials that will meet actual and future demands and future context also with global change. And of course, there is many things we have to revisit a lot, all the silviculture that used to be done before because of all these changes. So we need to have to increase and have a more effective knowledge exchange about planted forest, because the decision making is more and more complex and require more exchanges. Project websites often close at the end of the project, so we lose a lot of information coming from interesting projects such as Beforest, Homed, and so on. Scientific papers often do not focus on practical issues and so are not appropriate for managers, for stakeholders. Reports are produced in national languages and they are not often disseminated, even if there is some interesting input in them. Books are sometimes not republished. There's a lot of things in literature. So what we want to set up is something dedicated to people working on planted forest with all kinds of resources, including videos, guidelines, fact sheet, but it can be also tools, decision support system, any kind of data set that is relevant. We want to target practitioners and decision makers to facilitate exchange of knowledge between this group and the scientific community working hard on this topic. We want to focus on applicable methods, tools, recommendations, things that really can be used for any kind of decision makers. And we can try to, we want to organize it in a simple way so that it is easy to find. And we want to have this information secure on a long-term perspective, being an association where all the people contributing can be part of the governance. So that's it. Let's up to shape this future website. And there is a Clarkson survey open if you want to contribute. And thank you for your attention.
Thank you, Christophe. I would just like now to ask both panelists who are in the room and all the speakers to come up here. It's lunchtime now. We started a little bit late, but if there are, please come up here. I want to thank you properly. So, I don't have any more questions for you, but I really want to thank you and also our panelists online and the two speakers online for taking the time, for sharing your views, for sharing your research. I think we were successful in raising a little bit of awareness on forest genetic resources. Do you know more about forest genetic resources right now than you did in the morning? Yeah. Okay, great. So we achieved our goal.